and be so glad to have the opportunity to present our research to you. Mesenchymal stem cell, MSC for short, are a type of multiple lineage somatic progenitor cell with strong immunomodulation. Until now, over 500 MSC clinical trials are ongoing, and over 40% of them are used on immuno-related disease. This table lists MSC sources on these clinical trials, but little of them succeed. One of the reasons is because the different tissue source of MSCs. Until now, it's still unclear whether this tissue-derived MSC exert equal immunomodulation ability or not. For example, B cells develop in the bone marrow, which is also the first site where MSC were found. Does the origin affect bone marrow-derived MSC on B cell modulation still uncleared? From our study, we found that bone marrow MSC do not alter the B cell proliferation after stimulation. However, placenta MSC significantly decrease the proliferation. From testing figure for each B cell subset analysis, we found that, compared with stimulated B cells, bone marrow MSC support B cell further differentiation into pre-GC-like B cells. However, placenta MSC maintain B cell in a less differentiated state. It means more transitional B cell be visualized. Next, the lower right total cell count figure show that bone marrow MSC support B cell proliferation and their further differentiation. But placenta MSC suppress B cell further differentiation through suppression overall B cell subset proliferation. Next, we analyze another B cell subset we called BRAC cells. We found that bone marrow MSC do not alter this BRAC percentage, but placenta MSC increase this BRAC percentage by almost 30%. In order to test our in vitro findings are physiological relevant, we use an in vivo mouse inflammatory models by LPS inject intraperitoneally. We found that adoptive transfer bone marrow MSC do not alter the increase of B cell number and each B cell subset percentage. However, placenta MSC significantly suppress B cell further differentiation through suppress overall B cell subset proliferation, as well as increase the percentage of T1B cells with the same trend with in vitro result. Moreover, for the terminal effector plasma cell are assayed, placenta MSC showing significantly suppressed effect on it. Next, we also assayed BRAC subset, as we did in vitro. We found that bone marrow MSC do not alter this BRAC percentage. However, placenta MSC significantly increase marginal zone BRAC percentage and increase T2 and ZP BRAC percentage in nearly significant levels. Next, we're using human transcriptomic array to assay the factor associated with B cell modulation, which have been mentioned in the literature. We found that the factor which inhibit B cell proliferation, plasma cell differentiation, GC differentiation, and promote anti-inflammation cytokine secretion are IL-1 beta, IL-33, CCL2, and IL-12. As we can see, the expression of this factor is higher in placenta MSC than bone marrow MSCs. Moreover, the factor CXCL12, which is related with B cell development, is higher in bone marrow MSC than placenta MSCs. Therefore, we suggest that the tissue-specific effect of human MSCs is multifactorial. Now, scientists have focused on the role of B cells on those prototypic T cell mediated autoimmune disease. This NEJM paper reviewed a critical role for B cell on multiple sclerosis. So we can suggest that B-cell interaction may be the key to MSC therapy success in immune disease. From this study, we know that all MSCs are not equal, so choosing the right tissue MSC is critical. This study was recently published in Stem Cell Translational Medicine this year. Thank you very much.